Welcome back. The next speaker is Professor Eugenia Lo. Her talk is titled "A Breakthrough is Plasmodium vivax in invading human erythrocytes, how they get spread in Africa." She's assistant professor in biology science at UNC Charlotte, and he and his research focuses on molecular epidemiology, host parasites, interaction, and evolutionary genomics of Plasmodium, mostly in African countries. The talk will be in English, and she will receive questions in English. Please type your question to the speaker using the chat. Welcome, Professor Eugenia. You can start whenever you want.
Thank you for everyone and the organizing committee to invite me. Hold on, start my video. To invite me to this meeting that I can share with you all some recent work that produced from my lab, my team, and also my collaborator. As Jason said, my lab mostly focus on malaria epidemiology in African countries and host parasite interactions. And one of our key interests is to really understand this new phenomenon of how vivax malaria spread around African countries, as well as how they invade Africans, which is previously thought to be rare or absent in Africa. And this project is a highly collaborative project that involves multiple institutions. So I want to take a few seconds to thank everyone upfront before I forget. So this is a project that involves the team from Jimma University from Ethiopia, led by Dr. Daniel and our field team and technicians there. Also a collaborative project with Dr. Dan Janis from Bioinformatics. The team from Botswana European Partnership, we collaborate also with renowned cell biologist Louis Miller from NIH, with Sapienza University in Italy, Evangelical University of Cameroon, as well as the team led by Dr. Mazamu from University of Khartoum. Last but not least, of course, my team at UNC Charlotte Biology, which is fantastic, and they are really excited to be involved in this project. So malaria is vector-borne infectious disease, and we have been talking about this like over decades, which is still at this point not eradicated. And this disease is mostly caused by the parasite Plasmodium, transmitted by Anopheles mosquito from one person to another person. And if you look at the world distribution of malaria today, you can see over 90% of cases and deaths happen in African countries. And only like very tiny portions, 2% in South America and 7% in Asian countries, still affected by malaria. Just last year, and this number stay more or less the same, about 230 million cases and more than 400,000 deaths were reported. As I say, this number is more or less the same in the last decade, and we're still not doing enough, still not having enough resources to eradicate this disease. So malaria, in order for malaria to happen, it always have these four major different components to put in place. Of course, the first is the host. We human as the reservoir for the pathogen, but it also happens in a wide range of animal hosts like the non-human primates, in birds, in vertebrates, other invertebrates, etc. Right now, there are 6 major Plasmodium species, predominantly Falciparum and vivax, but also other minor species like Malaria, ovale, and simium that can cause sickness in humans. And all these 6 species, they are transmitted with by about 30 different Anopheles species, but the most common one in African countries is Anopheles gambiae, arabiensis, phanensis, and coluzzi. And when the host presence, vector presence, and agent presence, it still require a perfect humid, hot, watery environment in order for mosquito to breed and transmit from people to people. And this is how the best recipe for malaria to occur. And if in terms of vivax malaria, if you go back to 10 years ago in 2010, you will see, like, look at the distribution. Most of them happen in South America as well as in Asia. Only a very tiny portion of them happen in Africa, and they're mostly confined in the Horn of Africa, in East African countries, like Ethiopia, Sudan, Madagascar. And the reason why African population sort of immune or susceptible to malaria 10 years ago is because most Africans, they are Duffy-negative, which means on the surface of their red blood cell, they don't have this Duffy receptor that vivax parasite will need to use to bind and dock on the red blood cell before invasion. So that's why the distribution of malaria, of vivax malaria, is tightly related to the distribution of Duffy-negative around the world. But fast forward 10 years later, we are seeing an increasing number of vivax cases across different parts of Africa, which we previously thought or conventionally think is not possible. Just because these people, they are Duffy-negative, how can they infect by malaria? But we are seeing all these reports of symptomatic cases, asymptomatic cases, which can still transmit from one person to another person, not only in East Africa, but also in West and Central Africa. So the question is why? So if you look at the parasite invasion mechanism, it's a pretty complex process that involves multiple steps. Once a mosquito bites you, inoculate the parasite into your bloodstream. The parasite will produce these that have entered the first stage called erythrocyte that navigate around, find a healthy red blood cell. Once it locates a healthy red blood cell, it reorient itself perpendicularly to the surface of the red cell, forming a junction to the surface of the red cell membrane. And at this junction, you will see the parasite will produce a ligand protein that recognizes the host receptor, we call it a DAR protein, in order for to initiate the invasion process, which is to fuse the red cell membrane that allow entry of the parasite. So in other well-studied models, such as falciparum and nosai, we know there are multiple parasite ligand proteins that are labeled by the red box here that can be recognized by a number of host cell receptor represented by the yellow box here. But in vivax, so far we know that there's only one DBP, one protein, Duffy binding protein, that recognizes the DAR receptor on the human red cell for entry or for invasion. So in Africa, thanks to the great work by Louis Miller back in the 80s, in the early 80s, he recognized the fact that a lot of Africans, they cannot be infected by vivax, or there's a rare vivax malaria in African population. And what later on he found that is because their DAR receptor has very little to no expression. And therefore, vivax malaria, even though they can produce DBP protein, they just don't have the lock for their key to enter the red cell. So knowing the increasing number of cases of vivax malaria across different African countries, of course, there are three novel and important questions from the public health point of view, as well as from the scientific point of view, mechanistic why, how that happened. So the first question is, what are the epidemiological and genetic features of these infections, specifically in Duffy-negative individuals? Second, mechanistically, how they infect the Duffy-negative red cells. And lastly, at the public health setting, how these vivax malaria transmit from individuals, and what are the factors that govern infection transmission. And of course, because we only have limited time here, so I'm going to go over the first question, the epidemiological and genetic features. So in the last 3 to 4 years, in collaboration with teams from Cameroon, Botswana, Ethiopia, and Sudan, we've been able to collect a total of 14,033 febrile patients from different hospitals, including the one in Cham, Cameroon, from 2 different districts in Botswana, 3 districts in Sudan, as well as 2 districts in Jimma. And for all these febrile patients, what we did is we collect the thick and thin smear so that we can identify whether we see the vivax parasite in the red cell under microscope as our first line diagnosis. And then in the meantime, we also collect dry blood dot that obviously preserve the DNA of the parasite so that this sample can ship to my lab, and then we can do all the molecular screening for both the parasite as well as the Duffy status, whether they are Duffy-negative or Duffy-positive. And in order to unravel the genetic relationships where these vivax malaria come from or how they are interrelated among the African isolates, we also sequence the DBP gene and analyze them together with other published sequence deposit in the gene bank from other African countries, including 31 from Uganda, 4 from Madagascar, and 1 from Mauritania. So here are the results we are seeing. So what you are looking at is the different donuts with different color. So in Cameroon, if you look at the donuts, you see majority of our sample, among 217 samples, 215 of them are Duffy-negative. So the blue color represents the CC genotypes, which is Duffy-negative, okay? And then if you look at the sample we have from Botswana in Southern Africa, you will see only about 83% of them are blue Duffy-negative, but yellow, the rest of them are yellow, which represent a CT heterozygote Duffy-positive, as well as a red TT homozygote Duffy-negative, Duffy-positive. So this proportion, 83% compared to Central West Africa, say in Cameroon, is much less. But the most drastic difference we are seeing in terms of the Duffy distribution is between the East Africa and West Africa. If you look at East African countries like Sudan, Ethiopia, you are seeing only about 20 to 36% of our febrile patients are Duffy-negative. Majority of them are Duffy-positive. So there's certainly a huge difference in terms of the Duffy status distribution across African countries. Why? Well, it could be scaled back to like very historic human movement, migration, as well as population admixture reasons. So back in maybe like 1,000 years ago, the Bantu tribe from Central West Africa, specifically between Cameroon and Nigeria, these people, they are majority or predominantly Duffy-negative. It's possible that they will move southward to Southern Africa and mix with the Khoisan tribe that have a mix of Duffy-negative and Duffy-positive. So apart from the southern migration, they might also move to the East Africa. And as you know, most East African, they also have this Eurasian ancestry. So majority of these people, they also have a very dynamic, diverse genetic admixtures, and most of them are Duffy-negative. So when you have this Bantu that carry the Duffy-negative genes that move to the east, move to the south, mix with the indigenous Duffy-positive, that explains why you are seeing such a contrasting pattern of Duffy genotypes across across Africa. So now, knowing the background of the Duffy distribution in Africa, of course, the next question is, okay, among all these populations, what's the rate of infection when it comes down to vivax malaria? So the top panel here just show you the some of the numbers we collect from our patient sample. In Cameroon and Botswana, if you look, among all the febrile patients we identify as Duffy-negative or a small portion as Duffy-positive in Botswana, only 13.8% of these patients in Cameroon have vivax. And likewise in Botswana, as low as only 3% of these people are infected by vivax. And the rate of Duffy-negative in vivax, the rate of vivax in Duffy-negative, however, can go up to like 90% here, or 80% more. Now, if you look at the number we see in East Africa, you will see this number dramatically lower. So for example, in Ethiopia and Sudan, although the infection rate of vivax goes from 7.5% to as high as 37.4%. Among all these vivax infection, Duffy-negative people only accounts for about 3.8% to the highest 14.3%. So what you're seeing the pattern here is, if you look at the column, the number in this column, the last column, although East Africa has a higher rate of vivax malaria, you see a majority of infection in Duffy-negative. You will see that more or less in West Africa and Southern African countries, but not in East Africa. That means it's very likely that the parasite have evolved its invasion mechanism in West Africa instead of in East Africa, knowing the majority of the cases originated from there. And if you also look at the parasitemia, this also tells you something quite interesting. So although the different countries, they all show different rates of vivax infection, they have different rate of Duffy negativity. There's one consensus that we see across the board, which is the difference in parasitemia. So the plot, the scatter plot you're seeing here shows you the comparison of parasitemia by molecular screening across the vivax infection from Cameroon, Botswana, Ethiopia, Sudan. And for each country, we also compare them between Duffy-negative and Duffy-positive. So what we are seeing here is vivax density is significantly lower in Duffy-negative than in Duffy-positive infections, regardless of geographical ethnic boundaries. And this difference could imply that vivax in Duffy-negative, they may have a lower invasion capability. That's why they never can infect that much red cell to result in a high parasitemia. And among all the infections that screen positive by molecular method, for example, in Ethiopia, we found that out of 20 Duffy-negative vivax infection, only 3 of them are microscopic positive. As I mentioned earlier, microscopy is the first line diagnostic tool for our staff and technician in hospital setting to identify these rare, uncommon infections. But knowing that a lot of these infections are submicroscopic, which means definitely we need a much more sensitive diagnostic tool to allow us to vigorously screen and identify these infections in the field so that we can do more molecular, deeper assay in the lab. So to further understand the genetic relationship among all these African vivax isolates, we also use the DPP gene to construct a phylogeny with all the different African country vivax. So as you see, the green, the bubble here represent the Duffy positive, the square here represent the Duffy negative. So this phylogeny basically split into 3 major clades, 1, 2, 3. And what we are seeing is that Duffy negative, they do not form a monophyletic clade or cluster together. Instead, in each of these 3 subclades, we're seeing the square, the Duffy negative, always cluster together with the bubbles, the Duffy positive, without any geographical or country boundaries. That suggests that maybe all these vivax are basically circulating or sharing the same DPP haplotypes without clear geographical or Duffy differentiation. But of course, this is only this phylogeny is only based on 1 single DPP gene. There's still a thing that we don't know and also need to investigate further. Right now, this phylogeny will only allow us to generate hypotheses that's suggesting maybe the Duffy-negative and Duffy-positive vivax isolate, they are just coming from different origin, independently evolved multiple times. but still we don't know how they are related to the West African isolates, as well as what about to those Central American, as well as, South American, as well as the Asian vivax isolates. And more broadly, all the clades we are seeing here are only from the human reservoir, but vivax certainly are found in non-human primates. So would there be any genetic similarity between these two hosts, and whether the one in human hosts originated from apes long time ago? So all these questions require further phylogenetic analysis, as well as molecular dating analysis. And that's why right now, one of my students that me and Dr. Janice have been co-supervising, Anthony. So we just recently published a paper that published the first genome of vivax from African countries, specifically from Ethiopia. We identified over 120,000 SNPs that allow us to go further to use this SNP and build a whole genome phylogeny in order to thoroughly examine the question of where the Duffy-negative vivax originated from. So this is the current work that Anthony has been analyzing with almost 100 more genomes that we collaborate also with Wellcome Trust Sanger Institute. And apart from the description, the genetic description, knowing how much variation we are seeing at the genome level, my student Daniel is also now looking into the functional aspect from the transcriptomic analysis. We are trying to compare between the in vitro cell culture vivax as well as the in vivo human infected sample to see what are the genes that are highly expressed when these Duffy-negative individuals first infected by the parasite. So these might tell us something, what genes are really operating, playing the key role in invasion. And my another student, Corinne here, she's also doing a whole bunch of in vitro binding assay early this year to verify the functions of certain candidate genes. Hopefully in the end, we have more answer to what are the molecular mechanism that explain invasion. So this is my last slide, just to wrap up what I just talked about. Hopefully there's something that you guys can take home with. So based on the study we have so far, we now understand, know better that although vivax is prevalent in East Africa, the Duffy-negative infection is much higher in West and Central Africa. And we know also Duffy-negative infections always have a lower parasitemia, and they're mostly submicroscopic. So we definitely need a much more sensitive and rigorous screening tool to identify this infection. Genetically, Duffy-negative vivax in Africa, they are not monophyletic, but this remains as a hypothesis to test further by whole genome phylogeny and molecular dating analysis that will give us better insight into the timing of divergence and relationship, etc. And with all said, I think I'm going to end my presentation here and happy to take any question. Thank you.
Thank you very much, Dr. Lu. That was very interesting. Mainly, I, as a South American person, I find it very interesting because malaria is a problem. While we wait for questions, that I invite all the audience to feel free to put in English or in Spanish, and I may be able to translate them on the chat. I wanted to ask, being this a health issue, how can science approach it and help the politics and the social strategies to approach this very important health issue?
So one thing obviously is always about diagnosis and treatment in the clinical setting, right? And all these will need resources from the local governments, from broader agencies like WHO, to really put in resources, especially in more rural, remote areas, to provide them or train them with expertise. How to really diagnose this infection accurately, and also how to treat them in time, just because specifically related to vivax malaria, there's always this relapse. So if you don't treat them timely, that might affect you lifelong. So I think from as a scientist, obviously, is to make sure we produce this data and be able to present in more public setting to really convince the public as well as government agency that this is a serious issue that is affecting people may not be in the city, but like in the, as I say, in more rural setting. So yeah, that's why we need to talk more with all different type of kind of people.
Yes.
Thank you, Doctor.
I have another one as the mosquito. Go, Jason.
No more time, Lara. Thank you, Dr. Eugenio, for your talk and your presence today. And to participants, thank you for your presence. And we remind you that the next talk is now. Please check the calendar or on the Discord channel called Lobby Icebreaker. Obrigado por terem vindo. A próxima palestra pode ser encontrada no calendário. Por favor, entre no canal de Discord. E é isso. Obrigado. Thank you so much and see you soon.
Thank you.
Thank you, Dr. Lu. Very interesting. Bye, everybody.